Thanks so much. Okay, so yeah, my name's Lorna. I am in the third year now of my PhD based at Bangor University. Um, the title is Setting Thresholds for Good Status in Marine Ecosystem Management. So I thought I'd basically kind of just go through the PhD as a whole, seeing as I've got nearly a full hour, which seems like a really long time to be presenting for me. So I'm part of the summer CDT, which is the Sustainable Management of Marine Resources Centre for Doctoral Training. And I'm based at Bangor University with my primary supervisor, Professor Jan Hiddink. But I'm also working with Abigail McQuarter-Scollop at Plymouth, Tom Chagner at Exeter, and then as part of the CDT, we're kind of partnered up with different kind of organizations as well. So I've got Seb Valanco at ICES, Christina Herbon at JNCC, and then Sally Kaiser and Ian Gloin-Phillips at NIRAS as well. So a really great supervisory team. And I thought I'd just start off because for me, 45 minutes seems like a really long time. So I thought I'd just start off by just saying a bit about myself as well. So I didn't start off in kind of marine sciences. I actually did a psychology undergraduate in London. And then after that, I moved into qualitative research in London as well. And then I kind of had a kind of a COVID reevaluation of what I wanted to do really, and decided to do a marine biology master's at Essex University, which then led me straight into my PhD, which I'm going to be talking to you about today. So I got a picture of London and a picture of the Menai Bridge and the Menai Strait, which is where I'm usually based now. So quite a big difference, but I'm really enjoying being at Bangor. It's a really, really beautiful landscape there as well. So yeah, and I think the PhD really kind of combines some of my experiences with psychology and qualitative research as well, which I'll be talking about towards the end of the presentation. So just a really quick overview of what I'm going to be talking about. So I'll go through a quick background on thresholds and what they are and why I think they're really important. And then I'm just going to kind of take you through each chapter. So firstly, evaluating the different methods that are used to estimate thresholds. Then actually some work I've just finished, which is actually trying to estimate thresholds using kind of empirical data. And then the last chapter, which is kind of more on the qualitative side, using an expert elicitation approach to ask experts to estimate these thresholds. But I'm still working on the kind of analysis for that. So yeah, just some preliminary stuff there. And then I'll just kind of summarize everything and talk about some of the conclusions for that as well. So first of all, what are thresholds? Well, in the kind of context of my PhD, I'm looking at them as these values that distinguish between what is a good state and what is a degraded state. And you can see this figure on the left, which is from one of Jan's papers in the last couple of years. It's kind of this point at which we're defining when actually is good. And this can look quite different across kind of different indicators and different systems and even under different objectives as well. But what is this point at which we can define good and not good? And the reason these are kind of so important, as I'm sure many of you guys will be aware, is that there's lots of different marine legislations at the moment, which kind of have these good condition objectives within them and kind of looking at trying to achieve these different variations of what good is. And it's kind of, so if we look at the Marine Strategy Framework Directive and the UK Marine Strategy, we've got good environmental status by 2030, the areas beyond national jurisdiction, kind of looking at that more around the high seas. The Kunming-Montreal Global Biodiversity Framework, again, restoring degraded systems into something that is restored. And then the EU Nature Restoration Law as well, which is looking at restoring kind of degraded or poor condition systems into a good state. But it's actually quite difficult to try and actually define what is good and how do we know where to kind of draw that line and kind of put that zone there. And that's why we can use thresholds to distinguish between these different states. And under some of these as well, thresholds are required as well. So that's why we need them. But often they're actually not there, they're not in place, so we can't actually kind of assess the status. And it's quite difficult as well to really understand what method we should be using to kind of fill some of these gaps. So I think the PhD is really trying to cover some kind of answer some of these questions and hopefully try and provide this kind of a framework to give a bit more direction as to exactly what method we should be using and when. And that's going to be hopefully covered by these 3 chapters here. So evaluating the methods, setting thresholds, and asking experts. So the first chapter, evaluating methods, which was recently published actually. So please go and have a look at it, which I'm very excited about. So evaluating methods for setting thresholds for good status in marine ecosystems. And what we're trying to do here is really start to think about how we can evaluate different methods and what criteria we should be looking at evaluating these different methods against for actually estimating thresholds to try and understand, what one should we be using. So this kind of really began in the literature review, which was looking at and kind of other reviews as well. There's been some kind of ICES reports that are looking exactly at evaluating these methods against how statistically robust they are, how ecologically meaningful they are, and whether they incorporate some of these social and economic aspects as well, to try and kind of have this output of a framework as to try and understand, when we have this type of data, we should be using this method, and etc. So I kind of, as I mentioned, there's some kind of reviews in place already, which alluded to kind of how ecologically meaningful different methods were and how they kind of incorporated some of these social and economic aspects. But there wasn't really a lot of information as to how statistically robust different methods were in response to different kind of types of data. So when we're thinking about kind of statistical robustness and what we want to evaluate methods against within this kind of particular criteria, we're thinking about what type of pressure state relationship we're going to include, the level of noise, and the different sample sizes as well, and how the kind of different methods would respond to that and how those thresholds would be different. And so I looked at 4 methods. 2 of them use this reference condition data, and then 2 of them use the kind of data which looks at the pressure and state relationship. So I've got range of natural variation and statistically detectable change here. And range of natural variation uses, well, they both use this reference condition data. So theoretically data from an undisturbed area that has been kind of monitored over time. And therefore, we can see how this indicator, what the natural variation is if it was in an undisturbed and therefore good state. And the range of natural variation takes a lower percentile of this full range and says that's the kind of threshold, that's the lowest point that we can say that that system is still in a good state. And here we said the 5th percentile. And then you would apply that to the state in an impacted area to assess whether it was kind of above or below that threshold. Statistically detectable change, as I said, uses the same data, but instead of kind of looking at that full range of natural variation, it takes the confidence interval. So it's slightly different. It takes, theoretically, you could use different confidence intervals, but we use the 95th confidence interval, so the lower 2.5 as the threshold there, and then the same process. The second 2 methods use pressure state data. So tipping points and distance to degradation. So pressure state data, what I mean is basically how the ecosystem state varies as a function of the increasing pressure there. So what does that kind of curve look like? And for the tipping point method, we essentially look to find the maximum curvature on that pressure state curve and define that as the tipping point, the point before the ecosystem tips into a degraded state. And then distance to degradation, slightly different relationship here because we can't define a tipping point if the pressure state curve looks like this convex shape here. So you still find the maximum curvature on the pressure state curve, but that would be defined as the degradation point, because theoretically the system's already degraded by the time it's got down to that maximum curve there. And then the method says to, in order to define the threshold for good status, to basically add a percentile of between the maximum state value and that degradation point, basically just to push the threshold up the curve there. And that percentile would vary depending on the sensitivity of the system. So those are the 4 methods. And then the next step was to actually simulate data rather than using kind of empirical indicator data in order to control for these different variables that I wanted to see what effect they had on the thresholds. So I had 3 pressure state relationships. So a linear straight line, that sensitive, that convex curve, and then the tolerant, the concave one, and then the reference conditions as well for those other 2 methods. I then varied the level of noise in this data and varied the sample size. So this is an example of what one of those sample sizes would look like. So you've got, this is the largest sample size, which I think was 80. So you've got, each row is a different pressure state relationship, like I mentioned. So you've got sensitive, tolerant, linear, and then the reference conditions down the bottom. And then each column is a different level of noise that's been added to the data. So you can see the relationships really clearly on the left. And then as you move towards the right, you can see that they're super noisy there. And so what we did was fed this into these 4 methods to estimate thresholds on this simulated data. So this is an example of that simulated data I just showed you with the thresholds that these methods have estimated. So if I start with the pressure state data, so you can see straight away, obviously there's no thresholds for that linear relationship because there's no curve on that line there. The sensitive and the tolerant data sets, those rows there, they do have thresholds estimated. So if I move my window, distance degradation in purple and then tipping points in blue. So you can see, you can kind of see that even across the different levels of noise, the thresholds are quite similar, sitting at, I think, 0.6 or something like that at the top. But as we get further and further along, as the data gets noisier and noisier, the thresholds are not being estimated because it's getting so noisy that it's unable to fit that curve and therefore unable to kind of find that tipping point or that distance to degradation angle, and it can't fit the threshold there. And then if I go down to the bottom, so you can see statistically detectable change in red and then range and actual variation in green there. And what you can see, first of all, is that there's thresholds estimated for every single one of those data sets, no matter how noisy they are. What you can also see, I think green was maybe not a great color to use here, but for range and actual variation in green, you can see as it gets noisier and noisier, the thresholds are getting really, really quite low. Obviously, because it's taking the lower 5th percentile there. So as the data gets noisier, it's assuming all noise in this data is natural variation. So as it gets noisier and noisier, the thresholds are getting lower and lower and lower. And then kind of similar thing happening for statistically detectable change, but perhaps just not so much. And so what I did was, or what we did was just run that 100 times for each of the data sets and then collate the thresholds together in a figure, this figure, and basically showing you what I've already said. So as the data gets noisier, you've got the level of noise on the x-axis there. And I should say the way to interpret these thresholds, so they sit between 1 and 0. 1 would be your kind of carrying capacity, desire, kind of ideal desired state. And then 0 would be kind of, there's basically no state at all. So as you can see, as the data gets noisier and noisier, the thresholds are getting lower and lower, particularly for range match variation. For distance degradation and tipping points, they've got these numbers on the actual figure here. And that basically represents the proportion of thresholds that have been estimated out of 100 runs. So if you've got 1, that's 100 thresholds been estimated 100% of the time. And as you move from 0% all the way to 90% added noise, you can see that those proportions are getting much, much lower. So, 70 to 90% added noise, you're getting 10% of the time or less than 10% of the time thresholds are being estimated because of that noisy data. What you can see, the thresholds that are being estimated most of the time, this is the largest sample size, but most of the time they're quite similar across the board. So across the different levels of noise rather, which is interesting. And then also looking at, so that was noise at a fixed sample size. This is a sample size at a fixed level of noise. So, you know, the smallest to the largest sample size on the right-hand side. And what you can see here is that this really affects statistically detectable change. So at small sample sizes, the thresholds are getting much lower and much more variable. And that's because the method uses confidence intervals, which, you know, as you get smaller sample sizes, those confidence intervals widen, and the thresholds, the lower confidence interval is going to get much lower. This doesn't have as much of an impact on range of natural variation. And then for distance degradation and tipping points, the proportion of thresholds that are being estimated are really quite low. And the variability of the thresholds being estimated is increasing a lot as well. So you can see on some of the kind of tipping points and distance degradation on the very far left-hand side with the really smallest sample sizes, they're getting much more variable and also starting to produce impossible thresholds as well. So it's theoretically implausible to have a threshold greater than your carrying capacity. So it's also kind of not producing some useful thresholds there as well. So that was the kind of the results from that first chapter. And just to summarize that, those pressure state methods, or the methods that use pressure state data, they do produce kind of more accurate thresholds in that they're similar thresholds if they are being produced with kind of high sample size and that kind of lowish level of noise. They are kind of more accurate and they're quite consistent, but they're not very reliable in the sense that a lot of the time thresholds aren't being estimated, or they're quite variable as well. Whereas those methods using reference conditions, they're always estimating the threshold no matter what. But there's quite clearly a relationship between the level of noise and the sample size with both the range of natural variation and statistically detectable change there. So we kind of concluded that if the data is quite limited, so if it's a low sample size or if it's quite noisy, it's more reliable to use those methods with reference conditions, because we can quite clearly see the relationship between how these different variables of the data impact the thresholds, but they're always going to actually estimate the threshold as well compared to some of the methods that use pressure state data. So that was the first chapter. The second, or chapter 3, the second data chapter, I have been working on, and I've just kind of finished the kind of data analysis, and we're writing this up at the moment, which is really exciting. And this basically looks at trying to use some of those methods from that chapter I just spoke to you about and apply them to empirical data sets to actually estimate some thresholds. So I looked at using the reference condition methods. So when I say reference conditions, this is kind of long-term monitoring time series, minimally impacted system. I know I said undisturbed, theoretically undisturbed before, but I think in reality that's quite unlikely to find data that kind of meets that definition, and therefore we use minimally impacted. But essentially we're looking for long-term time series that show what the natural variation of the indicator is over time, kind of around this mean. And then using these two methods that I already mentioned, so range of natural variation, using that 90th percentile of the full range, and then statistically detectable change using that 95% confidence interval as well. So both those two methods are essentially what I use to try and apply to actually estimate thresholds to empirical data sets. And these are the long-term kind of monitoring time series of reference condition data sets that I managed to get my hands on, thanks to some co-authors and thanks to some other people that I will give a shout out to afterwards as well. So we wanted to find kind of indicators of biomass, density and abundance to kind of keep it quite straightforward. So we used plankton life-form abundance from the Continuous Plankton Recorder Survey around the North Sea and the North Atlantic, and we looked at 3 different life forms. And we used abundance data from the 1960 to 1980 because that was before these kind of quite widely acknowledged regime shifts to stay in line with that minimally impacted definition. We also had data from shoot density in seagrass, Zoster marina seagrass beds around the UK from the Isles of Scilly and Skoma. And that was from 1996 to 2023, which is amazing. And again, minimally impacted, speaking to experts that monitor those sites. We also used data from tropical reef fish that was collected by NOAA. And this was across 4 kind of broad trophic groups based on diet. And there were quite a few islands that they monitor, but we only used the ones that were uninhabited to try and remove as much as we could that fishing pressure that's associated with those. And that was from 2009 to 2023, but there were the actual number of sampling occasions weren't as many for this due to kind of interval sampling and then some impacts from COVID and things as well. And then we also had data sets from benthic invertebrate biomass species and then communities which were separated. And this was from some amazing work, mostly from some amazing work that some previous master's students spent a long time collecting from literature searches. I also had data from the Merman repository, from some people at Bangor, and then from some people at Natural Resource Wales that sent me data as well. And we kind of tried to ensure that there were only data sets with minimal physical disturbance, mostly associated with fishing pressure as well, looking at kind of Global Fishing Watch, talking to the experts who had these data sets, and then looking through the papers that contain the data as well. So that was all the kind of empirical data that we had. And then I thought I'd just go through an example of exactly how we would estimate a threshold using these data sets. So here I've got blue crab density. So this would be a benthic invertebrate species reference condition data set. So this is from 1990 to 2020. And what we would do to apply these 2 methods would be, we would standardize the data, and then we would detrend it as well. So we want to remove any shifting baselines, because although these are really important for kind of defining these absolute reference values, what we're looking at is defining relative natural variation around a mean. So we want to look at that annual variation. And so your kind of mean value here would be 1. So that, again, that's your kind of carrying capacity or your desired ideal state. And then we're looking at the deviation down from that. So applying these 2 methods, the lower 5th percentile, the lower 2.5 confidence interval, would give you a threshold of 0.77, so 77%, or 0.87. So that's how we would kind of actually estimate those thresholds. And that's what we did for all those data sets that I just showed you. And that's shown in this figure here. So if I take benthic invertebrate biomass and look at the community data sets on the left-hand side, the box plot here is essentially showing, I think it was about 33 data sets we had. So that would be the spread of 33 thresholds calculated across different data sets for the benthic invertebrate community biomass. And what you can see here, so we did benthic invertebrate biomass, we looked at the plankton life form abundance, the tropical reef fish, and then the seagrass. And then each of those are kind of community species, then large copepods, meroplankton, small copepods, the different trophic groups and shoot density as well. And then range of natural variation thresholds are in red and statistically detectable change in blue. So each one of those groups has 2 box plots as well to show that. And what you can really see is there's quite a bit of variation across the thresholds, across these different data sets within these kind of species indicator groups. Apart from seagrass, which I only had 6 data sets to be fair, but most of them were from the Isles of Scilly. So basically all those thresholds sat between 0.8 and 0.9. So there was really not much variation there at all. Compared to where you look at the kind of the benthic invertebrates, where I did have a lot more data sets, but there's quite a bit of variation there. And then I mentioned that there was not as many sampling occasions from the tropical reef fish monitoring data sets. And that's where you can really quite clearly see the statistically detectable change thresholds are much lower than range of natural variation. As we saw in that chapter before, if the sample size doesn't kind of meet this, particular like minimum value, then the thresholds from statistically detectable change are going to be much lower, just because of those confidence intervals. And then kind of on the other hand, you can see the plankton life form abundance, the range of natural variation thresholds are quite a bit lower there. And that's because there was so much variation in the kind of abundances of those plankton life forms as well. So what we wanted to do after doing this was to try and present this in a way where we're looking at kind of the distribution of thresholds rather than kind of a single kind of mean value across these different species groups. So we used these, we calculated these density distributions of the thresholds kind of within these different species groups. So I've just shown here the benthic invertebrate species and the benthic invertebrate communities. And what we can use these for, so the mean values of those solid lines, and then the most frequent values of those dashed lines there. And what we can use these for is to actually estimate the probability of a system being in good status, rather than kind of the, as I mentioned, the absolute mean value. So for example, if you had if your kind of impacted system had a state value of 0.71, you would look at this distribution and find that value and find that that would equate to a 50% probability of your system being in a good status. If the state was lower than that, at say 0.45, there would be about a 20% probability that that system was in a good state. And then vice versa, if there was a much higher state value of 0.95, according to this distribution, this spread of thresholds we have, there would be about a 90% probability that the system was in a good state. So that's a really, we thought that that was a really nice way of kind of presenting a bit more of the error associated with some of these thresholds and allowing decision makers to perhaps determine at what probability they think is kind of appropriate for defining thresholds for good status there. The second thing we did as well was to try and actually understand a bit more about whether we could explain some of this variation in threshold values I showed you even with those box plots. So again, I've just shown the benthic invertebrate species, otherwise I'll be here for hours. But looking at trying to explain some of the variation in these threshold values, we looked at the depth of the monitoring site, the sediment height, and the lifespan of the maximum average lifespan of the species that was being monitored. And we basically found that depth and lifespan significantly predicted the range of natural variation threshold that was associated with it. So, and that's what's shown in this plot here. So you can see lifespan on the x-axis, and then each of these lines is kind of a different predicted depth. And it basically says that the species with greater average lifespans have higher thresholds, and species at greater depths have higher thresholds, because there is less variation in their kind of biomass or abundance over time. So again, that's kind of a nice way of also potentially allowing thresholds to be predicted for species of a particular maximum average lifespan or at a particular depth, where perhaps the data or the reference condition data isn't available there as well. So just some conclusions to wrap up this chapter as well. What we showed was we basically estimated the thresholds for good status using these methods, allowing kind of people to actually estimate the probability of impacted systems being in good status. And we've kind of found these relationships between thresholds and environmental characteristics and the life history traits of the species, which can be useful for actually predicting thresholds if the data isn't available. And hopefully kind of setting out this framework that can be applied to other indicators as well, not just the ones we've included, if the data is available, to potentially kind of try and fill some of those gaps in assessment where it's applicable. And yeah, as I said, it took me a really long time to come up with some of this data. So I just wanted to give a shout out to some people that gave me some amazing data sets. And I really, really appreciated that. Okay, and then finally, I don't have as much to say about this one because I'm still working on it at the moment. I still have about maybe just under a year of funding left, so I've still got time to go through this one. But this is kind of separate in a way, I guess. It's not following on from these other chapters, but expert elicitation is often used to actually estimate thresholds. Not only when there is potentially a lack of kind of quantitative data for some of those methods I mentioned before, but also when perhaps there is needs to be a bit more of a nuanced kind of decision making, and to try and estimate thresholds where we perhaps don't even necessarily have the kind of the more beyond the data as to actually the theory and the knowledge available to actually come up with a value or a threshold where we just don't have one yet. And I think when I was kind of doing the literature review as well, it was often hard for me to find a kind of step-by-step kind of record of how some of these thresholds had actually been decided upon. Because if there is this kind of unstructured approach being used, we can't really replicate what's been done. We can't update it if we've kind of redone it and wanted to add new quantitative values to some of these structured methods. And we also can't evaluate the biases that are potentially in play when there's a kind of an unstructured elicitation method being used. So the idea behind this was to try and apply a more structured approach, firstly to understand how applicable it would be for estimating thresholds for good status, and kind of compare that to some of the quantitative thresholds that we've come up with. And then also actually to estimate thresholds to see kind of how and whether they could be used for these specific systems we were looking at. So the structured elicitation approach that we looked at was the IDEA protocol. And this is kind of based on the Delphi approach, which is really, really commonly used, really widely used, and essentially involves multiple iterations of a survey. So you would send out a survey to experts, they would fill it out, send it back to you, and then you would kind of compile those, and experts would be able to see anonymous responses from everyone else in the group. So they could kind of reevaluate their own kind of thought processes and opinions, and then they would carry out the same survey again and update and change their estimates if they wanted to. And the idea behind that is it kind of improves accuracy in their own estimates by kind of encouraging them to reevaluate what they've done, and to, yeah, I guess kind of have another kind of structured step-by-step process in how to do that. And I don't know if it's necessarily the most common approach, but the 4-step elicitation approach is used. So I'll show you an example of what I used, but it basically gets experts to, as well as asking them, you know, what do you think the threshold is? It also is, how confident are you in some of the estimates you've given? And the idea behind that is to kind of encourage them to kind of reevaluate what they've done, and that has been shown to reduce overconfidence in estimates as well. And then the reason that this is always done as an individual exercise and before it's ever brought together as a group is to try and completely remove that groupthink bias, which can often happen if you get a bunch of people in a room together. It can often, instead of encouraging people to have their own thought processes, it can often encourage people to, obviously think as a group, which is not necessarily always bad, but if you want that kind of individual variation in estimates to get more of a spread, then yeah, you are, I guess, removing that. So the IDEA protocol only differs from the Delphi approach in kind of one aspect, really, is that it includes a group discussion in the middle of kind of when you give when you send the survey out to experts. So they still see other experts' estimates anonymously, but they're not anonymous because they're talking to each other as well. So the idea behind this is to reduce some of the ambiguity in the language that's being used in the survey to encourage kind of a similar interpretation of what the question is actually asking. So show you some examples of what I did. So we chose to look at seafloor integrity, which is descriptor 6 on the in the MSFD in the UK Marine Strategy, specifically for benthic bivalves. And this was honestly really because the majority of my supervisors are more kind of benthically specialist. So it was kind of chosen so that I would hopefully be more likely to be able to speak to enough experts to actually do this, because that's inherently a really difficult part of doing a study like this. So we looked at seafloor integrity, which is specifically looking at those benthic systems, and we looked at horse mussels, a general bivalve community, and a cockle bed as well. And we used case studies for each of those, and then had kind of 2 questions for each kind of case study indicator. One was looking at the quality, so the kind of density biomass, and then one was looking at the extent, the area of the system as well. So the examples I've included here are just for the kind of horse mussels. And so the first one is, assume that the density of a Modiolus bed in pristine condition is 600 Modiolus per meter square. What density of mussels needs to remain to avoid adverse effects on the structure and function of this bed? So that would be kind of the quality question. And then the second one is essentially the same, but is looking at the extent of the area as well, the system. And then this is what I was talking about with this 4-step elicitation approach. So experts get asked, what do you think the lowest plausible value could be? What do you think the highest plausible value could be? What's your most likely estimate? And then how confident are you that your lowest and your highest plausible value, that interval, captures the true value? So that's the kind of the survey that we used. And yeah, it was a really, really interesting study. So this is, as I said, this is really preliminary. I'm still looking at the results and I'm still analysing everything, but I've included some early stuff as well, so I have something to speak about. So so we've got the case study questions down the bottom. So there were 6 questions, right? So 2 for Modiolus, which I just talked you through, and they're very similarly 2 for a general benthic bivalve community and 2 for a cockle bed as well. And we gave experts kind of information about a specific case study that they could try and kind of ground the estimates in. And this is, so if you look at the third one, so realistically, what is your most likely estimate? That's what this figure is showing. So this is kind of a compilation of all their thresholds, their best guess, best estimate thresholds across 6 of those questions. And then the average is in blue there down the bottom. And I think what the kind of key thing here is that there's obviously a huge amount of variation in some of those thresholds. I think slightly less in the Modiolus extent, interestingly enough. But yeah, there's a lot of variation. And I asked some of the experts this. I included a kind of optional group discussion right at the end to see. It's not part of the kind of I guess overall structure of the IDEA protocol, but I kind of wanted to speak to them a bit more to try and understand a bit more about how they were interpreting the different kind of I guess definitions I included. And when we talk about seafloor integrity, and when I spoke about those questions in there, I was asking, at what point is the structure and function adversely affected? And this definition that's from the MSFD and the UK Marine Strategy of not adversely affected has kind of two kind of components to it almost. In green here, it says the ecosystem maintains its respective components, i.e. structure, along with its natural levels of diversity, productivity, and ecological processes functioning. But the second bit, it also kind of refers to recovery potential. It must be at a level that ensures dynamic recovery potential. And so I tried to go through and kind of pull apart whether I thought people were interpreting it one way or the other. And I think for some of them, it did explain a little bit more about the variation in some of those threshold estimates there. So particularly for the kind of general soft sediment bivalve community, that third one there, you can see it really kind of pulling apart where people were perhaps interpreting it not incorrectly either way, but just differently. And so that's really interesting, but I've only really been working on this last week as well, so still quite early. But it is interesting to see perhaps whether that could explain some of the variation in some of these estimates as well, and some of those interpretations. So some quick conclusions from this before I wrap everything up. As I said a few times, it's definitely still in progress, some of this analysis, so it definitely might change. But I think this was a really useful approach to try and understand a bit more about how we're defining what actually good is, and how that may mean slightly different things to different people. And I think you can definitely use this method to estimate thresholds where you don't have data available, or where you need a bit more of a nuanced approach to it. But I think it is obviously really important to try and make sure that the interpretations are the same of what you're using. And I think some of the lessons learned from me, and I think I spoke about this with some of the experts as well, is I think it would have been really useful to refine some of the questions with them and refine some of the case studies that I included with them. And then perhaps for the future, if I were to do it again, I would ask, like, what approach did you actually use to estimate the threshold? Because I think we did speak about it in some of the groups, but I think it would have been really useful to get their kind of thought processes down whilst they were doing it as well. So a really quick summary of the work that I spoke to you about today. So the kind of first chapter, we simulated thresholds to look at how statistically robust different methods were. And what we found was that methods that use these reference conditions, if there is limited data, they're much more kind of straightforward to understand and much more reliably going to estimate thresholds with that data. The second chapter I presented, we looked at kind of using a framework which uses these methods that use reference conditions to set thresholds to determine how we could estimate the probability of a system being in good status. And we found that we think this is quite a transparent, consistent, and quite easily understood approach actually to estimate some of these thresholds and hopefully fill the gaps where they are needed to. And then finally, the structural elicitation approach we use to try and estimate thresholds for seafloor integrity. And as I said, analysis is still ongoing, but potentially what is good varies slightly by expert and could depend on what approach is actually being used to estimate that threshold there. So yeah, thank you. That's everything from me. Yeah, love to hear any questions you guys had on any of that as well.